to my video lesson on plasma membrane, which is the gateway of cell. Dear students, you might have seen a watchman at the entry of the factories or at big houses. What a watchman can does, he cannot allow the entry of unauthorized persons into the factory or industry or any big houses. In the same manner, he cannot allow the carry out the materials belonging to that industry or factory or that house. It means he regulate the entry and exit of materials or persons from the industries or factories or at big houses. So he is regulating. In the same manner, there is a regulator in the cell. It is a membrane. It is called plasma membrane. Here also the plasma membrane is performing same thing as a watchman is doing. He regulates the entry and exit of materials. So it is a gateway of cell. So in this video lesson, let us discuss in detail about the plasma membrane. So now let us discuss the brief history of the plasma membrane. First, it was observed by Robert Hooke. It is believed that when he studied the cell wall, and he also observed the plasma membrane. So it is named as plasma lemma by a scientist called Flower, and it is named as cell membrane by Nageli. The scientist called Overton studied well the structure of the plasma membrane. First, this plasma membrane is present next to the cell wall. So in the plant cell is covered by cell wall. So next to the cell wall, a thin layer is present. It is called the plasma membrane. So it is present outside the cytoplasm or outside the protoplasm. So if we take one cell, in the center nucleus is present generally, and around the nucleus cytoplasm is present. In the cytoplasm, cell organelles are present. The cytoplasm and the nucleus together called as protoplasm, and this protoplasm is covered by plasma membrane, and this plasma membrane is surrounded by cell wall. So the protoplasm, which is surrounded by this plasma membrane, is known as protoplast. In mycoplasma and in animal cells, only plasma membrane is the outermost layer. In that organisms, no cell wall is present. So in that organisms, plasma membrane is the outermost layer. So plasma membrane is also called as cell membrane or plasma membrane. Generally, the plasma membrane is present attaching to the cell wall. So in normal cell, this plasma membrane can't be observed. This can be observed in plasmolized cells when there is exosmosis, when there is movement of water out of the cell, then the plasma membrane which is present next to the cell wall, by attaching to the cell wall, it moves inwards. So at this condition, when it moves inward, this condition is called plasmolysis. So shrinkage of protoplast is known as plasmolysis. When there is shrinkage of protoplast, then the plasma membrane which is attached to the cell wall, it moves inside. When it moves inside, then we can observe clearly this plasma membrane. But generally it is present attached to the cell wall. So when it is attached to the cell wall, it is not possible to observe clearly this plasma membrane. So plasma membrane can be observed clearly in the plasmolized cells. So plasmolysis can take place when there is exosmosis. When cell is placed in high concentrated solution, then exosmosis takes place. So at this condition, we can observe well this plasma membrane. So the chemical composition of the plasma membrane. So what substances are present in plasma membrane? Generally, in the plasma membrane, lipids, proteins, carbohydrates are present. Generally, in the plasma membrane, in the middle, lipid layer is present. On either side of this lipid layer, protein layers are present. Generally, the thickness of the plasma membrane is 75 angstroms, but it also ranges up to 150 angstroms. So when it is 75 angstroms, the middle layer, that is lipid layer, is 35 angstroms thickness, and the two protein layers which are present on either side of this lipid layer, each one is with the thickness of the 20 angstroms. So this is a general composition of the plasma membrane and general arrangement of these proteins, lipids. So carbohydrates are present by attaching to the proteins or by attaching to the lipids. So this kind of arrangement is there in the middle lipid layer and on either two sides, and on the two sides these protein layers are present. So let us discuss in detail the chemical composition of the plasma membrane. That means the lipids, about the lipids, proteins and carbohydrates. So lipids, the lipids ranges 40%. When we compare to the carbohydrates and proteins, this lipids occupies 40%. Remaining 60% is proteins and carbohydrates. So here in the lipids are four types. There are phospholipids, glycolipids, sphingolipids, sterols. So four types of lipids are there in the plasma membrane. So in these four, phospholipids are more in proportion. So the concentration of the phospholipids is more when compared to the other lipids. So phospholipid has two portions, like one head portion, one round head portion, and below this one tail portion, one rounded head portion, and below this one tail portion. The head portion is hydrophilic. It means it attracts the water, and this head portion is having the phosphate molecule, and below this head portion, tail portion is there, which is hydrophobic. It means it doesn't attract the water, it repels the water, it throw out the water. So it is connected with the fatty acids. So remember, here one head-like structure, rounded head-like structure, and below this one tail-like structure, and head-like structure is hydrophilic and the tail-like structure is hydrophobic, and head structure is connected with the phosphate molecule and tail structure is connected with the fatty acids. So this molecule has two properties. It has hydrophilic property and also hydrophobic property. So this molecule is called amphipathic. Amphipathic. So it has two properties. So it is called amphipathic. This lipid portion. So just now we discussed that in the center lipid portion is there, on either sides protein portion is there. So the lipid portion is acidic in nature and it binds with the protein which is negatively charged. So by binding with the protein layers on either side, this plasma membrane is formed. Proteins. Here three types of proteins are there in the plasma membrane. One is structural proteins, and second one is functional proteins, and third one is carrier proteins. So structural proteins means they are involving in the structure of the plasma membrane. So they are the building blocks of the plasma membrane. So these proteins, the structural proteins, are two types: peripheral proteins and integral proteins. So just now we discussed that in the center here lipid portion is there. So these peripheral proteins, they are present on either side of the lipid layer, but they are attached loosely with the phospholipids. So just now we discussed that the lipid has two portions, one rounded portion which is present upside, that is phospholipid, that is phosphate. So it is attached to this phosphate molecule, and these are present peripherally, not inside the lipid region. Whereas integral proteins, they are present inside the lipid region. Peripheral proteins are present on the lipid region. Integral proteins are present inside the lipid region. Sometimes they project out. Some portion of the integral protein projects out and they may be present on the margin of the lipid layer. So these are the structural proteins. So they are involving in the formation of the plasma membrane. So functional proteins, second type is functional proteins. So these are enzymes. We know that enzymes are proteins. So here the enzymes which are present in the plasma membrane are acidic phosphatase, alkaline phosphatase, and RNase. So these enzymes involve in the biochemical reactions of the plasma membrane, and they are useful in the performing the functions of the plasma membrane. And next one is carrier proteins. The name itself denoting that it carries the ions. It means plants absorb the materials, minerals in the form of ions. So that ions are carried into the plant in the form of ions, and they are carried by this plasma membrane and with the help of proteins present in the plasma membrane. So some proteins, they carry the ions present outside the plant, the soil, and also they are useful for the movement from one cell to the another cell. Carbohydrates, when compared to the proteins and lipids, they are very smaller in proportion. So just now we discussed that in plasma membrane, three types of molecules are there. They are lipids, proteins, and carbohydrates. But these carbohydrates are very less in concentration when compared to the other two molecules. And carbohydrates are sugars. So here the sugars, some sugars are branched form and some sugars are unbranched form in the form of linear form. And the carbohydrates are two types in the plasma membrane. They are glycolipids and phospholipids. So glycolipids and glycoproteins. They are glycolipids and glycoproteins. It is written wrong. So it is glycolipids and glycoproteins. So glycolipids means, glyco means carbohydrates. So the carbohydrates which are attached to the lipids is known as glycolipids. The carbohydrates which are attached to the proteins are known as glycoproteins. So some carbohydrates are binding with the proteins, they are called as glycoproteins. Some carbohydrates are binding with the lipids, they are called glycolipids. So when glycolipids are present in the plasma membrane, they are useful in the transmission of information to the cell. They also acting as antigens. The glycolipids also act as antigens. So whenever one foreign material tries to enter into the cell, if it is toxic material, then it identifies it. So this glycolipid identifies these toxic substances which are trying to enter into the cell, and it restricts these toxic substances not to enter into the cell. So the plasma membrane is made up of three molecules. One is proteins and lipids and carbohydrates. So the arrangement of three molecules is explained by different scientists with different models. But there are many models. But now let us discuss four mostly described, mostly accepted models. So these models explains about the arrangement of lipid protein molecules in the plasma membrane, and the carbohydrates are present linking with the proteins and the lipids. So Danielli and Dawson model is one model which explains the structure of the plasma membrane. In this model, in the center, lipid layer is present. In the center, this is the lipid layer. This is the lipid layer from here to here, from this yellow region to this yellow region. This is the lipid layer, and this purple color globular molecules are proteins, and here also proteins. So proteins, this is one layer, and from this yellowish round to this yellowish part, here this is one layer and this is one layer. So here three layers are present. So it is also called as trilamellar model. So lamellae means layers. So three layers are present. So it is called trilamellar model. And here two protein layers. This is one protein layer. This is another protein layer. These protein layers are sandwiched with the lipids. So you might have known this sandwich. When prepare the sandwich, two bread slices are linked with the sandwich, one reddish edible juicy like substance. So in the same manner, this protein layer and this protein layer, these two are binded or linked with the one, this is linked with the one fat layer. For example, this is protein layer and this is a protein layer, and this white one is the fat layer. So these two layers are sandwiched with this fat layer, so it is also called as sandwich model. See, in the lipid zone, the molecules are arranged in two rows. So bimolecular region it is. This is the one row of lipid molecules, and this also one row of lipid molecules. So lipid molecules are arranged in two rows. So bimolecular lipid region it is. So in one row, the lipid molecule has one rounded head. This is hydrophilic, which attracts the water, and this is the tail region. This is the hydrophobic, which don't allow the water, which throws out the water, which repels the water. And in the same manner, also here also another lipids row of molecules are present. So in one row, the tail regions are present here inversely in the another region, so that the tail regions in the two rows are arranged oppositely. So they are arranged in oppositely, and the head regions are away from the tail regions, and on either side these are the protein molecules. So this is the model proposed by Danielli and Dawson. And next to unit membrane model. This is proposed by Robertson. So he accepted the model of Danielli and Dawson. So he accepted the model of trilamellar, and he also proposed that not only the plasma membrane, all membranes present in the cell shows the same kind of model, shows the same kind of structure, so that his model is named as unit membrane model. So it is a unit, this structure is a one unit, so same kind of structure is there in all membranes, so it is called unit membrane model. So here, so the model is same as the trilamellar model or Danielli Dawson model. It is the center bimolecular lipid zone, on either side proteins are there, and here the proteins are fibrillar, whereas in the earlier model they are globular. Now here they are fibrillar, like this, they are arranged, and the external protein layer is mucoproteins, and internal protein layer is non-mucoproteins. And this external, this hydrophilic head of the lipids is positively charged, and this tail region is negatively charged, and this lipid region is binded with this protein region. So this is the model. which is proposed by the Robertson. Next to micellar model. It is proposed by Hillier and Hoffman. According to this model, plasma membrane is not lamellar model. It is not formed with the layers. Whereas earlier models explained that here the molecules are arranged in the layers. But according to this model, the molecules are not arranged in the form of the layers. So it is not the lamellar model. So according to this model, the plasma membrane is formed by globular units called micelle. So like this is one micelle and this is one micelle and this is one micelle. So with this micelle, this plasma membrane is formed. So the unit of the plasma membrane is micelle. And on either side of this micelle, protein molecules are present. So this micelle is formed with the phospholipids. It is formed with the phospholipid molecules. So in the phospholipid molecules are arranged in a circular form, and the hydrophilic head region is present away from the center, and hydrophobic tail region is present towards the center. So like that, one circular or globular structure is formed. So with these units, with the union of these micelle units, here plasma membrane is formed. So in between this micelle, the pores are filled with the water. That is explained by Hillier and Hoffman. So this is about the micellar model. Fluid mosaic model. It is proposed by Singer and Nicolson. This is mostly accepted model among all the models which are proposed by different scientists. Fluid mosaic model. So according to this model, the phospholipid region is in semi-fluid manner. In the semi-fluid, some protein molecules are floating on this semi-fluid phospholipid region. Some are also floating, but they are inside this lipid region. So when we see this plasma membrane with these proteins, some are on the phospholipid region which are floating, some are embedded in the phospholipid region. So when we see this one, it gives one mosaic appearance, and this phospholipid region is in semi-fluid manner, so that it is called fluid mosaic model. So here also the molecules are arranged. The phospholipid region structure is same as earlier scientists explained. Here also the polar head region is this outside. It means towards outside. It may be this side or it may be this side, and tail region is present towards the inside. So here this plasma membrane is not static. So according to this model, the protein molecules, they cannot stay at one point. These protein molecules, they move from one place to another place. So they are not in static position. In the same manner, the phospholipid molecules also, they move from one row of these molecules to another row. They exchange their positions. For it means this molecule can move to this one, at the same time this molecule can move to this place. So the phospholipid molecules can exchange their positions, and this tail region, it can rotate on its axis. See, this is the, for example, this is the head region, this is the head region, this tail region. This tail region may rotate on its axis like this. So here all the molecules are not in static condition. Wooden molecules can move from one place to another place in that semi-fluid lipid zone, and this lipid molecules also, they may exchange from one row of lipid molecules to another row of lipid molecules, and also the tail region can rotate on its axis without changing its position. So this can explain well the functions which are performed by this plasma membrane. So this is the mostly accepted model. Next, to discuss about the functions. Plasma membrane has one unique character known as semi-permeability. So permeability means it allows all materials. Semi-permeability means it allows only some materials. Impermeability means it can't allow any material. So these are three terms. Impermeability means can't allow any materials. Semi-permeability means allows only some materials. Permeability means it allows all materials. So plasma membrane has the unique character that is known as the semi-permeability. Due to this semi-permeability, only it is functioning the watchman duty to the cell. Now let us discuss the each function performing by the plasma membrane. Transport of water and solvents, and also gases. So the plasma membrane, it can allow only the solvent molecules into the cell. For example, when there is a solution outside the cell, when there is a solution outside cell, due to its semi-permeable nature, it allows only the solvent molecules to enter into the cell. It can't allow the passage of solid molecules into the cell. It allows the entry of the only solvent molecules like water or any other solvents. So this kind of process is known as end osmosis. End osmosis. Osmosis means movement of water molecules. Movement of water molecules from low concentrated solution to high concentrated solution through plasma membrane is known as osmosis. If it takes towards the cell, it is called end osmosis. If it takes place out of the cell, it is called exosmosis. If the cell is placed in high concentrated solution, then here exosmosis takes place. If it is placed in low concentrated solution, then end osmosis takes place. And next to ion transport. So remember, in the plant cells, the cell wall has the nature of permeability. It means cell wall allows the all materials to enter into the cell. But the plasma membrane which is present next to the cell wall has the character of the semi-permeability. So it allows only some materials. Ion transport. So plants absorb the minerals in the form of the ions. For example, if sodium chloride is there, when we put to the sodium chloride to the plant, then it dissolves in the water and it converts into the ions sodium plus and Cl minus. Then the plant absorbs these minerals in the form of ions. So this plasma membrane absorbs the ions in two methods. One is active absorption and second one is passive absorption. Active absorption means absorption of the ions with the help of ATP against the concentration gradient. It means when there is high concentration in the cell, for example, in the cell, sodium ions are more in number, and outside the cell, sodium ions are less in number. So, sodium ions are entering into the cell towards the high concentration. So, they are entering into the cell against the concentration. So, they are not moving towards the low concentration. They are moving towards the high concentration. So, they are moving towards the high concentration. Then they need some energy, so they enter into the cell with the help of the ATP. So one more time, you listen that movement of ions against the concentration gradient with the help of ATP is known as active absorption, and passive absorption, it is just reverse to this one. Movement of ions according to the concentration gradient, and first one is against to the concentration gradient, and second one is according to the concentration gradient without the help of the ATP. It means the ions move from high concentration to low concentration and without the help of the ATP. This is the passive absorption. Passive means no any utilization of the energy. So passive absorption and active absorption, these two processes takes place through the plasma membrane. And here in plasma membrane there are some ion carriers are there. Ion carriers. These ion carriers, these are proteins, they are present in the plasma membrane, and they carry the ions from outside to the into the cell and from inside to the outside of the cell. So this ion carrier transport system is of two types. One system is called uniport and another system is called symport. Uniport means here movement of single ion, only one ion. Symport means here movement of two ions. So uniport and another one is biport. Movement of two ions is biport. Movement of single ion is uniport. So biport is of two types, symport and antiport. Symport means movement of two ions in the same direction. For example, movement of these K plus ion and also Cl minus ion in the same direction. Movement of K plus ion and Cl minus ion in the same direction into the cell. This is the symport. And antiport means if the K plus ion moves into the cell and Cl minus ion comes out of the cell, this is called antiport. So here one is uniport and second one is biport. In uniport only one single ion transportation is taking place. In biport two ions transportation is taking place, and it is of two types, symport and antiport. In symport two ions are moving in the same direction. It may be into the cell or out of the cell. And in antiport the two ions are moving in opposite direction. One ion is moving into the cell and one ion is coming out of the cell. And third one is endomitosis. Endomitosis means intake of large molecules. And in the same manner, exocytosis means exit of large molecules. So exit of large molecules out of the cell is known as exocytosis. Intake of large molecules into the cell is known as endomitosis. Endomitosis is of two types. Pino, N O should be there. It is it is wrongly written. So PINO, pinocytosis. Pinocytosis means intake of large liquid molecules. Phagocytosis means intake of large solid molecules. So intake of large molecules, endomitosis. If they are liquid molecules, it is pinocytosis. If they are solid molecules, it is phagocytosis. So pinocytosis means cell drinking. Phagocytosis means cell eating. Cell eating is phagocytosis. Cell drinking is pinocytosis. So when there are large liquid molecules outside the cell, then the plasma membrane, then it surrounds that large molecules, and slowly that move into the cell, that engulf into the cell. So here the plasma membrane slowly moving outside, and it is surrounding the molecules, whether it is maybe solid molecule or liquid molecule, and after its surrounding, then that molecule that slowly absorbed into the cell. In some cases, that is taking place with the help of receptor. Receptor molecules are present in the plasma membrane, and these receptor molecules and the molecules which have to enter into the cell are known as ligands, and these receptors, they bind with the ligands, and after binding, slowly the ligand and the receptor, they diffuse into the cell, and after entering into the cell, then the ligand is pinched off and they move into the cell, and again that receptor moves towards the outside of the plasma membrane. Then again it is ready to carry the another ligand. So exocytosis means it is movement out of large molecules. And special functions, microvilli. So in the small intestine, their microvilli are there. Microvilli are formed with the plasma membrane. When plasma membrane is projected out, and then this microvilli are formed. Due to the microvilli, then only the intestine, in the intestine, the absorption capacity is increased. See, the plasma membrane present on the intestine towards the inside of the plasma membrane, it is projecting towards inside. So the area of this plasma membrane is increased due to formation of microvilli structures. So this microvilli, due to presence of microvilli, then the intestine absorption capacity is increased. The area of the plasma membrane is increased, and so that the absorption of the digested material is increased. And pellicle. The pellicles are present in the protozoans, which are useful in their protection. So antigens are present in the plasma membrane. Actually, they are glycoproteins. So whenever a foreign toxic material tries to enter into the cell, it identifies that material, then it triggers the formation of the antibodies. Then these antibodies they fight with this toxic foreign material, and it cannot allow the entry of the toxic foreign material into the cell. And these glycoproteins are also useful in the transport of hormones into the cell, like insulin hormone. So, due to which this sugar disease, diabetic disease is taking place. WBC, white blood corpuscles. The white blood corpuscles they are surrounded by the plasma membrane. This plasma membrane it identifies the pathogenic bacteria and also kills the pathogenic bacteria. So that only whenever there is a infection in a human body or any organism, there the WBC number increases to fight with the pathogenic bacteria. So this action is taking place with the help of the plasma membrane only. So in this video lesson, the students we have discussed about the different components of the plasma membrane and their arrangement in the plasma membrane, and which are the different models of the plasma membrane and which are explained by different scientists. So here we have discussed four models of plasma membrane structure, and also now we discussed the different functions of the plasma membrane. I think you got this lesson and you understood well this lesson. Thank you and best of luck.